Hi, my name is Liz DeMarco, and I'll be talking about B cell development. First, we must discuss hematopoietic stem cells, or HSC. These cells are found in the bone marrow and begin differentiation. Stromal cells are also found in the bone marrow to support the development of cells into B cells. Stroma is a Greek word for mattress. These are supporting cells, and they signal B cell differentiation. As B cells begin to mature, or cells that are going to be B cells, they progressively have less and less contact with stromal cells. They begin to move towards the central axis of the bone marrow and eventually leave the bone marrow altogether and go out into the periphery. VCAM1 is a type of cell adhesion molecule from the stromal cells to promote binding and encourage proliferation. IL-7 encourages proliferation and growth. A signaling molecule, FLT3, is secreted from stromal cells and encourages the differentiation of the stem cell into a CLP. As a CLP, the cell can continue to become any type of lymphoid cell. Now you might wonder how the CLP will commit to a B cell. IL-7 and stem cell factor are secreted by stromal cells, and this encourages the CLP to commit to a B cell. The cell then enters the pro-B cell stage. In the early pro-B cell stage, the cell undergoes rearrangements of the heavy chain locus. The cell will first open up the heavy chain locus and go to D to J recombination of the heavy chain. RAG is present, or RAG1 and RAG2 proteins, and these allow the cell to begin the VDJ recombination. TDT is also present and promotes junctional diversity. Once the D to J recombination is complete, the cell moves on to become a late pro-B cell. In this stage, the cell undergoes rearrangement of the heavy chain into the V to DJ recombination. The rearrangement of the heavy chain is therefore completed in this stage. RAG is still present. If the rearrangement is unsuccessful, it will attempt again at the second locus. If this is unsuccessful, the cell will therefore die. If the rearrangement is successful, it will enter the pre-B cell phase, under which it will go proliferation. The cell produces a surrogate light chain, which is a fake generic light chain that is uniform throughout the pre-B cells. It is used in place of the real light chain to determine if the heavy chain that was just produced is successful. The expression forms the pre-BCR complex. If this is unsuccessful, the cell will die. The pre-BCR complex is expressed on the surface of the cell to signal that the cell is ready for the large pre-B cell stage. The complex includes two proteins, Ig alpha and Ig beta. These proteins signal to stop VDJ recombination. Therefore, it stops expression of the surrogate light chain RAG and TDT. This promotes allelic exclusion. In other words, only one heavy chain will be expressed on the final BCR. The cell then enters the large pre-B cell stage. In this stage, there is proliferation of cells that have the same heavy chains. No RAG is expressed during this stage to ensure that there is no premature recombination of the light chain. The proliferation amplifies the successful recombination events because it increases the population of cells with the productive heavy chains. The cell then enters the small pre-B cell stage. RAG's levels increase once again to begin recombination of the light chain. The cell is now considered a small pre-B. The cell does V to J gene recombination on the light chain. The kappa locus undergoes recombination first, and if that is successful, it recombines at the lambda. The cell will form the BCR complex, and the light chain will pair with the successful heavy chain. They will come together once again with Ig alpha and Ig beta proteins to form a mature BCR complex. This signals to stop recombination at the light chain locus. Once again, we have allelic exclusion to make sure that only one heavy chain and one light chain are expressed. IgM is now expressed on the surface of the B cell, and the cell is now an immature B cell. The immature B cell undergoes negative selection. Before the immature B cell can leave the bone marrow, it must go through negative selection to make sure the BCR is not self-reactive. If the BCR is self-reactive, it will go under receptor editing and the B cell won't be able to be released into the periphery. It will go back and try again at the second locus. If this is unsuccessful at both the kappa and lambda, it will die, and this is known as clonal deletion. If the cell does not react with its self-antigens, it is known to be self-tolerant, and then expresses both IgM and IgD on the surface. Now the cell is an immature B cell, and it will go out into the periphery and enter a secondary lymphoid organ, which could be the spleen, lymph node, or mole. This is where the immature B cell finishes development to become a mature B cell. Cytokines are released and are cell survival signals, and this will ensure the cell becomes a mature B cell. When the mature B cell goes out into the periphery and it encounters an antigen, the cell will then further develop into either a plasma cell or a memory cell. Thank you, and good luck on the exam tomorrow.